Okay. I just want to ask us to just spend a quiet moment thinking of everybody that is affected by the COVID-19 pandemic. Just a few moments in silence to just remember them, as well as all the healthcare workers. Okay, thank you very much. I'm Dr. Utilitarian. I'm from South Africa. I am a nurse by profession and I'm a senior lecturer at University of South Africa. It is my privilege to welcome everybody this morning to this conference and taking your time off from a very busy schedule. Welcome once again, and I hope we're going to have a very fruitful session during this conference. And I send my warm welcome to all participants, students, and delegates for the Scientific Global Digital Gathering at UCG 2020. I just want to tell you a little bit about UCG conferences. Utilitarian Conference Gathering offers a wide range of events, meetings, conferences, workshops, and symposiums. We have a dedicated team who aim at acquiring the technology, adapting the knowledge of business needs, and accelerate with best ideas and strategies. Utilitarian Conferences Gatherings is a pioneer in the area of event management and collaboration. We are full service events providers specializing in the field of medical, clinical, healthcare, life sciences, engineering, pharma, environmental science, engineering, business, agricultural, and food event services for academic and industrial sectors. We stand out distinctively from our competitors for our commitment to quality, round-the-clock service, and unmatched price. We are able to provide you with the best event, meeting experiences under one roof. We at Utilitarian Conferences Gathering make sure that you, as a client, choose the most appropriate event. I thank you all for finding time in your busy schedule to be here today. Today marks the major activity in the brief existence of the UCG conference, a moment where we are interacting, sharing experiences, and reflecting on various areas. I will now hand over to the chairperson, Mr. Ian Tonga from UK.
Thank you so much, Dr. Telly. I am what can best be described as a long-term practitioner in the art of successful management of diabetes for my life, to which I'm going to be presenting this later. However, my role here is to welcome you to the UCG 2020 conference. And just explain a little bit from my understanding as well. UCG 2020 is a remarkable event which brings together researchers and students in the field of healthcare, making the webinar a perfect platform to share experience, operations across industry and academia, and evaluate emerging technologies from across the globe. We cordially welcome you join us and witness the advances in this field together. The conference has attracted delegates who have come from all over the world, across continents, and the subject of the conference is surely of global in UCG thanks all individuals and organisations who have worked to make this conference a grand success. UCG Programme Committee have put in long hours and many creative efforts. And finally, I would like to extend my appreciation to the participants of this conference. We hope you enjoy the conference and that our participation will contribute to a mutually professional development of relationships. Can I now pass back to Dr. Telly?
Thank you very much, Mr. Ian Tonga. I'm going to introduce to you now the keynote speakers. I take the immense pleasure of introducing these speakers at the UCG 2020 conference. The first keynote speaker I'd like to introduce is Dr. John Shillingford, Avon Technology from UK, Professor Tarek Abdel Hadi Ali from Cairo University, Egypt, Ms. Amal Ibrahim, Al Najah National University Hospital in Palestine, Ms. Hand Abdullah Al Jafar, First Health Cluster E1 Eastern Province region from the Kingdom of Saudi Arabia. Then we've got Mr. Syed Al Nobani, Cleveland Clinic, Abu Dhabi in the United Emirates. No? Then we've got Dr. Salman Al Marzouki, Devo Team Group from the Kingdom of Saudi Arabia. Then we have Mrs. Judith Amlas, Center for Grateful Leadership, USA. And we have Dr. Alan Nazar Stakos from Australia. I thank you very much. And now we will start actually with the beginning of the keynote forum. And it is my great pleasure in calling upon the keynote speaker, Dr. John Shillingford from UK to deliver his keynote speech.
Okay.
Dr. John Shillington, are you ready to share?
I'm sort of ready. I'm just trying to see how this technology is going to go. First of all, thank you for inviting me. I must say I started my career before computers were even going, and when I wrote my PhD up, it was on a typewriter. However, here I am all these years later on a virtual conference with people from all over the world. So it's a great honor. So thank you very much for inviting me. It's been really, it looks like being an interesting couple of days. All right, now, shall I begin by getting my screen up and sharing that, okay? Okay, let's do this. Right. Now,
Dr. Shillington,
is that
while you are
sorry?
While you are preparing to share, may I just mention you've got, actually we're starting a little bit earlier,
okay.
But you've got 30 minutes to do your presentation.
Okay.
And may I ask the other delegates to not interrupt the presentation. If there's any questions, they might pose their questions on the Q&A. But it looks like there's no Q&A button here. So I suppose we just carry on. And with the permission of everybody, sorry, can you kindly ask your permission to record this meeting, this gathering?
Okay. Now, can everybody see my screen?
Yes.
In which case then, hey, that's the first problem I had I was concerned about. Okay. Right, first of all, my name is John Shillingford. I have been in the industry, as I've said, forever in terms of pharmaceutical and medical device development. I started in 1974, so you can now work out how old I am vaguely. Okay, it's a long time ago. I am the director of a company in the UK called Afon Technology. I've spent my life in, as I said, clinical research. I am, however, presenting this from my basement office in my home in Gothenburg, Germany, where I've lived for the last 20 years. I've been involved with AFON from the very beginning in their search for a non-invasive glucose measurement technique. Okay, so let's just go. All of this, you know, a lot of people are way better than me on diabetes. I am, after all, originally a biochemist who moved into organizing clinical research, and I've organized clinical research all over the world in various different projects. I've lived in the States as well. Okay, just talking quickly about diabetes. We know what the challenge is, and we know what the function is in terms of needing to control the disease. Okay, so self-monitoring is the big thing, and this is where we're going with the new technology that we are developing. We know that over the last few years, there's been a lot of technology that has been developed. Talk a little bit about that, but I want to talk about the future, which is where we are intending to move to. Okay, so here we are monitoring the hyperglycemia, hyperglycemia, and also we're now to the actual individual diabetic monitoring their own blood glucose levels as much as possible. And then, of course, the latest, which is looking at time in range, i.e. blood glucose levels between something like 70 milligrams a deciliter up to maybe 170, as being a key parameter in managing the disease. What we need is devices, technology to actually support the patients through this. Of course, what happens is as soon as you're developing devices, and this has happened over the last 10, 15 years, and that is the regulators come in and quite rightly start waving their wands and saying, okay, it has to be within certain parameters, which are, of course, safety for the patient, and also the data that we're producing has a total integrity. So what are we looking for? What are the regulators saying to us? Okay, you want to produce a new technique, a new method of using blood glucose. These are the parameters that we now have to operate to. So we're talking about, you know, we need to produce something which is 95% of the glucose results within 15 milligrams per deciliter for glucose levels less than 100 milligrams per deciliter. It's worth saying at the moment that the finger prick method, which we'll talk about in a minute, is usually around about plus minus 15 milligrams per deciliter. So if you're producing a new method, you're producing a new device, these are the parameters you need to operate on. And of course, the thing is that once the blood glucose goes up above 100 milligrams per deciliter, and actually way up into the 200s, it actually becomes, in our experience, more difficult to accurately measure. So we're really looking at, in developing a device, we're looking at both ends. We want to look at the low, the 50 milligrams per deciliter, i.e. when you're going hypoglycemic and it's dangerous, and then we want to look at the high, going up above 200 milligrams per deciliter, which is also not exactly the place to be. The other thing the regulators are looking at is something called the MARD, which is the mean absolute relative differences. It's basically an overall measure of how accurate your device is in measuring blood glucose compared to the gold standards that we all have to pitch our development programs against. And the other thing is there's this wonderful Parkes error grid, which gives a lovely picture, and we use this, but we need to be in what they call zones A and B, and I'll show you what I mean here by this photograph here. This data, okay, where you want, okay, on your left-hand side here, you've got the blood glucose, which has been measured by the device using the algorithms. Now, we'll talk about methodology later. And then the bottom bit, you've got the reference blood glucose, which is your gold standard. I can talk about that. What you want is to pitch your points right in the middle, going up, you know, in the dark green and the light green area. Once you start wandering out of that into the yellows and the reds, then you're really producing results which are not going to be acceptable to all regulators. Point is this, and that is, this was our first ever first ever proof of concept study that we did on 6 patients in a clinic here in Germany. It was the fact that it was a very scattered indicates, of course, it was an early stage of the device development. But we looked at it and we went, well, it's not bad. There's got to be something here. In actual fact, the people that we worked with in the clinic looked at the data, looked up. We thought it was too scattered to really be reasonable. And a couple of the professors looked up and said, you know what, I think you've got something here, guys. That was a year ago. Okay, here we go. This is for the standards. This is plus minus 15 milligrams per deciliter. For those of you who are diabetic, you know very well you do this a few times a day, and after a while your finger gets a bit sore. Although you tend to find, as I do, my thumb, which in actual fact is pretty pain-free anyway. Okay. The gold standards that the authorities tell us that they want to see against. There's the YSI machine. The Americans, the FDA, like a gold standard. Anything you're producing, you want to go against the gold standard from the YSI. But here in Europe, we tend to use the Super GL from the Müller-Gerätebau. And they are, you know, those are the standards that we've been using in our research projects. Again, accuracy is important for the gold standard. So what have we got on the market at the moment? We've got a couple of invasive devices which are basically dominating the marketplace. So there's a Dexcom and Dexcom 6, and there's the Abbott Libre Freestyle. Both systems basically sample interstitial fluid. So they're looking at the glucose levels in the interstitial fluid of the skin. So you are having a patch which is put onto your, usually the forearm, and they've got micro needles in them. That patch lasts about 14 days. They're not cheap to buy these things. And indeed, the performance can vary greatly from patient to patient. Although I have to say, over the last few years, the algorithms that they have built have become better and better. And the possibility of now doing continuous glucose monitoring is actually for real. And the acceptance by the FDA and by the Europeans that no longer do they have to put themselves as, shall I say, adjunctive to finger pricking. They get to now stand on their own as being standalone glucose measurements. So they're good systems. But the sensors are invasive. They are in the arm. They are microneedles. And we believe that a truly non-invasive approach is actually where we need to go in the industry. Just a word about the invasiveness. These continuous glucose monitors that use these microneedles, they do cause skin irritations and skin complaints. And a recent publication in the Diabetes Technological Therapeutic Journal has indicated that 18% of participants get skin irritations. Indeed, some of my colleagues that work with me in AFON who are type 1 diabetics using these do complain of that. And also 80% of pediatric users have issues. And also there's an issue there with some women in pregnancy as well. So those are the downsides of it. Emerging technologies, which I have to talk about, there is a lot, and the marketplace is really frantically working. This gives you an idea of the sort of stuff that's going on out there. You know, there's, again, microneedles, sampling interstitial fluid for Nomura. Medtronics have got an N-light system, which is again a microneedle inserted under the skin. Earlobes, interestingly enough, when we were starting on the journey of developing our microwave sensor, which I'll talk about in a minute, we tried earlobes, but earlobes are quite variable, and everybody has a different style. I think it's about 7 or 8 different types of earlobe, and the variation we found was quite huge. And if we were pinching the earlobe, of course, the blood left the earlobe itself, and so it became difficult. So I'm looking at that going, I wonder. Tear-based, well, you know, that would be something inserted in the lower eyelid, and so we go on. But then we move into things like low power laser. We're looking at, you know, infrared side, and also, again, GlucoWise is looking at low power microwaves. All right. Just a couple of things that are comments really about stuff that's being published at the moment. There was a study published recently using a multi-sensor devices, electrochemical spectroscopy. They had a MARD of 35.4, which is too high. It's as simple as that. You know, you want your MARD to be down below 15% approximately, but certainly below 20% and as low as possible. And interestingly enough, they published that. They're working on their device. It needs some more work, that's clear. Also, there's a I can never pronounce that. Anyway, whatever. The bottom line is, on that particular, they're looking at putting in monitoring using wavelengths of different sources, etc., etc. A very complex device. They had MARDs, which is the big measure, between 5.6 to 20.8%, which is actually very good. The issue there is that that's been done with volunteers using just a meal, so therefore their blood glucoses are within those magical 70 milligrams per deciliter, up to maybe 170. So they're really within almost normal ranges as such. And in our experience of research, we've found that to hit MARDs in those areas is actually relatively easy. It's when you start having data which is coming from more than 170 milligrams per deciliter, or below 70, down to 50 or 40 or 50. You don't want to go below 50 too often. But essentially, that is not as sensitive, and that gives you more problems. So I would imagine if they're going to take that device into a situation of looking at more extreme diabetics, their MARD levels will go up, from my experience. So what have we done? Well, first of all, I took a call from an American friend of mine who is a professor of statistics from Harvard. And he said, John, I guess you've retired. I said, no, not yet. I'm still working. He said, well, I said, I've got this mad group in the UK that need a clinical guy to come in and help on development. I said, yeah, okay. What is it? He says, it's a non-invasive glucometer. And I just went, yeah, right, you know. But anyway, I said, I'm in, because, you know, one likes a challenge towards the end of one's career. And I went in and I looked at other areas, looking at microwave technology, and I found a couple of groups there. One was in Cardiff, and they've been producing a device which basically goes on the abdomen, in actual fact. And then there's a group in Korea that have been looking at microwaves for quite some time, and they've done a lot of work in vitro and some work in vivo, but it's essentially the majority is in vitro still. So what happened here in AFON? Well, first of all, I joined the group. The work already that had been done was very much a sort of a, you know, early stage, in vitro, very much using dummy gels to mimic blood concentrations. But Sabi Chowdhury, who's the CEO of the company now, was the investigator up at the University of Liverpool. Looking at this, and he noticed that there were about, you know, there were about 4 or 5 resonances, you know, low-powered microwave spectrum, which will actually appeared to be sensitive to the different glucose changes in the bloods that he was messing around with. He actually managed to get samples of 10 subjects and do it that way. So the whole thing was that we were looking at a resonance shift of those 5 identified resonances within the spectral range that we're working in, both by moving in frequency, but also by amplitude as well. So the idea was that with knowing that and looking at that, if that is for real, then we can start actually building the algorithms. So in actual fact, we found a guy in the States, in Baltimore, who's a statistician and yet a signal analyst. And he's been working on the algorithms for us in the development of this technique. So what have we got? We've got a planar resonator, which is basically about the size of a €2 coin, or a, I forgot what English money's like now, a 10 pence coin. It's about that size, which sits currently on the volunteer's wrist, on the underside, not the top side. We've connected that up to a VMA. It looks like a Heath Robinson equipment. It's huge. But we took the study. We wanted to do the study in the UK, but there was an issue there. And we went in the end to a company called Profil, which is a diabetic experienced unit in Neuss and in Mainz in Germany. Why there? Basically because the whole of the pharma industry takes all their insulins there, and all of the drugs used to lower blood glucoses have been studied there. And these group have also seen every medical device in terms of glucose measurement that's ever been produced. So it seemed to me that the place to go. The other thing is that they can do the insulin clamping tests so that we can go high and low. We thought if we're going to go for this, let's really challenge ourselves. Let's just mess around within normal levels. Let's go and look at the high, look at the low, to try to see if we really have got something or if we have not got something. Okay, so we moved forward. We moved into a proof of concept study with first of all, 6 patients. I've shown you the grid in those lovely colors. But we then moved on from that, having been heartened. We went off and raised a little bit more cash from some investors. And we went back to Mainz, in actual fact, to Profil, to do a few more patients, okay, and to harden up our proof of concept data. Okay, so here we are. We went back and we did another 16 patients. We've done 6 already. And we started to look at MARDs, an average MARD of 22. Our aim was that we would progress the project if we reached about the 20-odd MARD. We actually reached 22.2. But what we really liked was we saw a range. We saw some really good MARDs of 12, 14, 15%, and some really high ones. Indicated to us that, you know, the device in some volunteers, or some volunteer patients, sorry, these volunteers were all type 1 diabetics, which would allow us to put them on insulin clamping to go really high and really low. And don't forget, those MARDs there are from really high and really low levels. So that is challenging the data. The other thing that then we saw, and that is that the strong correlation between the device algorithms and measurements that were coming out. And we were getting in the regions 48.9%, in the tight region 47.1%. So we were beginning to get into that 95% of the middle region on the grid, the lovely colored grid that I showed you earlier. So we were beginning to get there, and we began to think that really this was potentially looking okay. Okay, so just to give you a little bit of numbers, a little bit of a picture of what's going on. What we did was, and what we've always done, and we will be doing going forwards, is we had the patient for 2 days, and the blood levels that we measured on day 1 and the resonance changes that we saw on day 1, we would build a model, and then we would model that. We would then use that model on day 2 to predict the blood levels that we would see from the resonance changes that we would see. So basically, and then we would do the reverse. So basically what we're doing is we're building a model from 1 day's data and using that model to actually predict the following day. So you can see here that on the left-hand side, we've got the model with the predictions coming actual fact from the 2nd day with the actual blood levels, and then on the 2/16/08 October, that was the other one. That was the first day's model predicting from the resonance changes compared to the observed glucose. So that looks actually pretty good, you know. One swallow and a summer and all that business. At the same time, we ran in parallel Abbott Freestyle, and you can see the data below. In one case, it wasn't quite on the line. The other one looked pretty good. Okay. So, looking at that, you know, we've got a great model. We again, you can see from our lovely picture on the left-hand side that all of the little predicted and the actual blood are all within the lines that we're looking for. We've got a MARD of 12%, and that looks pretty good. We were very happy with that. However, as I always say, my years in clinical research means if I see data that's too perfect, I don't believe it. So, you know, it's inevitable then when we start looking at other cases here. This was the worst one we had, which is 32% MARD. And you can see in the lower levels there, the bloods, this is below the 60 milligram per deciliter, going into the yellows and the reds. So that indicates really that we've got very poor sensitivity down there. And this was my point, that we have to be you know, we have to look very carefully at the low and at the high ends. If we're going to produce, for instance, an indicator account device that just goes red or blue, or depending on where the blood levels are, then, you know, we need to be sure that if we are indicating, that we're indicating correctly. So therefore, you can see on the graph on the right-hand side, there appears to be some sort of, you know, the predicted effect doesn't match the blood lift, although it gets better during the day. Now, why? Very good point. And those are the questions that we have to ask ourselves in terms of the microwave device itself. Is it penetrating the skin properly? Was the device warming up? Was there something about the patient first thing in the morning that changes things? It's all about, I believe, it's a case of looking at the data, looking at the good data, but also looking very hard at the not so good data and analyzing why. Okay. And again, you know, another model indicating things weren't quite brilliant, but at the same time, we're getting there. We also, if we wanted an indicator device, then there's an indication here that there could be a future for an indicator device. So that would be sort of like a watch, which would go maybe green if everything's okay, and it might go red if you're high, or a different color if you're low, indicating that you need to go off and do a finger prick just to double check everything. Anyway, so where are we? Yeah, I mean, this is where we're going at the moment. We're micro-engineering everything. The guys back in Chepstow are a group of micro-engineers, software engineers, circuit guys, physicists, mathematicians, and a couple of clinical, but predominantly they're engineers. So we're in the process of producing that prototype at the moment, which we intend to start pushing into good human beings sometime in the next few months. Okay, so what have we got? Well, we think that we've got something. We think that the signal penetration from the microwave, which, by the way, everybody says to me, are you going to fry the skin? The power of the microwaves are far lower than those that you use in your portable phone, which sits in your pocket all the time. As we apply for regulatory approval to do these studies, and we have done with the MHRA in the UK and with BfArM in Germany, of course, they're hot on that sort of radiation. Just to reassure you all that they have looked at that, the output of that radiation, and gone, no, this is well within normal limits. So, what we are saying here, in that everybody's wrist is different, everybody's different. One of the wonderful things about being a biochemist, as I was many years ago, was I came to realize very early on that the human body, or the biological systems, are so infinitely complex that there's never black and there's never white. There's always shades of gray in between, and so that's where you get your variation from. And we need to look at that. We need to really profile what sort of patients, what sort of thing. You know, what changes do we need to do to the device to optimize the device to the individual? So that, a lot of work to do. I mean, basically a lot of work to do. And just like all good research projects, often these things throw up way more questions than they do answers. And for me, as a lifelong researcher, that's what I live and die for. However, you know, we do believe that we are moving very quickly. We will have the prototype device to actually go back in to run in large numbers of patients sometime towards the end of this year. Oh, hang on a minute. Right, yeah, okay. I've just forgotten where I am. Okay, so just a quick word about, you know, us. We've been funded by small investors, which has been great. And these investors have stayed with us over the last few years and have been absolutely brilliant in supporting us. And if you are an investor, then thank you so much. We operate on very strict, very tight budgets. We don't have big medical device or big pharma budgets. So everything we do is very carefully done. But the one thing we are insistent upon, and that is that we're going to work with people and centers that really have got the expertise and experience, And really, it's a case of us working nicely with Profil in Germany. We're also beginning to get under work with the University Hospital in South Wales. Excuse me, just a minute. Right, okay, let's shut that one on. And we're, as I said before, we're just about to go into more clinical studies towards the end of this year. Well, look, as far as I'm concerned, that's where I am, and that's all I've had to say. I wanted to point out that the whole research area in diabetes, in blood glucose measurement, is really going ahead very, very fast. Whilst our small group of scientists, technologists, engineers think that we have something which is interesting, of course, time will tell. One swallow doesn't make a summer, and we've got a long way to go. And I hope that as we move forward, we can come back to you sometime in the future and give you a better update on where we are. But for now, I'm going to say thank you, and I'm going to stop sharing my screen if I can do that. Stopping at the top. Okay. Right. Okay. Am I done? Hello? Can I dance now? Okay.
Unmute. Thank you so much, Dr. Shillingford, for a very informative and interesting presentation. And I think in many ways, we are all affected by the diabetic, being a diabetic, or else know of somebody who is a diabetic. Thank you very much for this presentation.
Absolute pleasure, and thank you for inviting me. And you know, I know that you're going to put shall I say, the presentation out on the web. It will have my name and address, and anybody who's got any questions for me, queries, or want to get involved, just send me an email and I will reply to everybody. Okay.
I just want to pose a question. If there's any of the attendees, because we've got some time left, if we'd like to ask a question, pose a question to Dr. Shillingford. You're welcome to do so. You can unmute.
Could I ask, Doctor, would it be possible for an individual such as myself to volunteer their presence in your prototype test?
Absolutely, yes, certainly would. I don't know where you're based, but you know, as I said, we're going to we'll be running a project down in just outside of Newport University unit there. Or contact me and then I can give you details. Okay.
Okay. Well, I'm in Manchester, so that's not really that near to Newport, but hey,
never know.
Put a card right away.
Chepstow's a lovely place. Come and see us.
Absolutely. Thank you.
Pleasure.
Thank you, Dr. Shillingford. Is there anybody else who would like to pose a question to Dr. Shillingford?
Okay. Well, thank you very much, and I shall go on mute. Okay.
Okay. Are you done? Thank you. If there's no more questions, then